video or year one video for A Level Biology. Today I'm going to be taking you through osmosis, quite a topic that some people find quite difficult. I'm going to do a separate video at some point on the osmosis required practical or PAG, but for this one we're just looking at how osmosis works. So osmosis, you need to understand about membranes to remind you about osmosis. So with membranes, don't forget the main structure of the membrane is the phospholipid. And you've got the phospholipid bilayer. So osmosis can happen freely through the phospholipids because water is a small enough molecule to fit through the gaps between the phospholipids. You may use something called aquaporins in some cells, such as the kidney cells and the nephron, because that is needing an extra amount of water. But for most cells, it's just moving through the phospholipid bilayer. So at GCSE, you would have described osmosis as this. You would have said that osmosis is water moving across boundaries, and it's the movement of water molecules from it's diffusion of water molecules from a dilute to a more concentrated solution, or you might have said, possibly from a higher to a lower water potential, which is what we're going to look at today. And it's always through a partially membrane. Don't write that definition down yet because we're going to look at a better definition later on. But basically what happens here is this. If you have a sugar solution, water will always move from the pure water towards the sugary solution. The picture's not very good. So let's have a look at it as you try and get a better definition. There you go, it's a better picture for you. So here we've got two different pictures. You've got a high concentration of solutes. The bigger molecules here are solutes, okay, and they are obviously maybe sugar or salt, something like this. You've got a lower concentration of solutes here. So we've got more free water molecules. If you look at this solute at the top left-hand side, these little molecules here are around the solute are water as well. When we say there's a high water potential, we mean there's more free molecules of water to the ones here. And water will always move from an area where there's more free molecules of water to an area where there's less free molecules of water. And that is because the water itself, the free molecules of water, are able to fit through the gaps of the membranes. So, how do we describe this in terms of osmosis? Well, that's an example second. Well, how we describe it is something to do with water potential. So water potential is the measurement of the potential that water will move in or out of a solution. And it mixes with a lot of things to do with pressure, concentration, things like that. But what you need to know is if something is dilute, then it has a high water or a higher water potential. Okay, and the highest water potential you can have is pure water, which has a water potential of 0. Okay, now that then will also link to then water will move from a higher water potential to a lower water potential. So something with lots of solute dissolved in it. So for example, glucose liquid or sucrose solution is often used in exams. That will have a lower water potential. Okay, so the more dissolved in it the lower things are. So we could reconfirm the osmosis definition now as the movement or the net movement, which means the overall movement of water molecules from a region of higher water potential to a region of lower water potential through a partially permeable membrane. And that definition you need to get into any exam question you ever asked about this. And don't forget, the purest water, so pure water, has a water potential of 0, which is the highest water potential you can get. Anything dissolved in the water potential lowers down and becomes more negative. So let's have a look at the examples. So this really shows you the 3 or 4 different examples you can have. So I suppose there's 6, okay? And they always ask you the same question. So all you've got to decide is it talking about an animal cell or a plant cell when it's asking you questions. So let's have a look at this then. So if we just look first of all at this, we've got water potential, okay, and we said water potential always moves from water potential the highest of 0 down to as negative as it goes. Okay, so we'll go down to like minus 100, minus 1000, whatever. Okay, so water potential, if the water potential is high on the outside of the cell, that means it will be lower on the inside of the cell. So water will move from a higher concentration in the solution into a lower concentration of the cell, and the cell will expand, swell, or burst, and we'll talk about that later on. If there's equal water potential on the inside and outside the cell, water will move both out and into the cell at the same level. So there's no osmosis because there's no net movement of water from a higher concentration, a higher water potential to a lower water potential. And that is because the concentrations are the same. If we've got the last example though, and there's a low water potential outside the cell, the water will move from the higher water potential in the cell to the lower water potential outside of the cell. So if we apply this to examples, we could sort of see that obviously it doesn't matter if it's an animal or plant cell, if you've got the same concentration, it's isotonic inside and outside, nothing will happen. But let's have a look at these two. So if we put it in pure water, which would be what we call a hypotonic solution, the water potential will be higher outside the cell than inside the cell. So water will move into both the plant and the animal cell because it's moving from a higher water potential outside the cell in the solutions. Let's say it's a sucrose solution, so you move from the higher water potential in the sucrose solution to the lower water potential in the cytoplasm of the cell. Now that will cause both cells to increase in mass, and if too much water moves in, the animal cell will burst open, which is called hemolyzed, and plant cells won't burst because the cellulose cell wall is too strong and therefore it resists the pressure. And at some point, the plant cell will no longer be able to fit any more water in, and it becomes like maximum volume. And we call that being turgid. Now, if the water in the plant cell goes the other way, so let's say you put it in a water potential of a low water potential, so let's say you put it in a very concentrated sucrose solution in a low water potential, the high water potential is in the cell, that moves out to the low water potential outside of the cell. Now that means that the cells, if it's a plant cell, it will come shrivel. Animal cell, it will shrivel up, which we call crenated. And the plant cell, the cell wall doesn't change because of the cellulose. But if you look here, the cell membrane shrivels away inside the cell wall. That is called being plasmolyzed. So it appears that the cell shrivels up inside the cell wall. So here we've got a common example that you might have. If you look at this graph, it shows you how many cells burst, what percentage. So at the top here, we've got 100% of red blood cells bursting in different salt concentrations. And we go from 0 salt concentration to higher. So basically 0, if you think about it, is the highest water potential, and then the water potential slowly dropping. So up until about what 3.25, the cells all burst because the water potential outside the cell is higher than inside the cell. So therefore the water moves from a higher concentration outside the cell, higher water potential, sorry, outside the cell, to a lower water potential inside the cell, causing the cell to burst. But then you see the concentration, the number of cells that are bursting drops. And if we wanted to know what the concentration of the blood cell is, it would be the point where 50% of the cells burst is what we call the isotonic concentration. So the concentration of this blood cell would be approximately 4, I'm guessing it's moles here because that's where it would go down to if my face wasn't on the screen. And then eventually the cells will stop bursting completely and they'll start shriveling up. And that is when obviously they're in a hypertonic solution, when the water moves from a higher water potential inside the cell to a lower water potential outside the cell. And here you have the different examples. So you need to know the 6 ways that things will happen. So again, water potential, if you put in a hypertonic solution, that's where you have a low water potential. Let's say, I'll just change my color. Let's say a low water potential might be minus 200. Okay, so the water will always move from a high water potential to a low water potential outside the cell. The animal cell will shrivel up. Okay, the plant cell will plasmolyze. Its cell membrane will shrivel away from the cell wall. In an isotonic solution, there's equal movement inside and outside because the concentrations are the same. There's no higher to lower water potential, so nothing happens there. And in a hypertonic solution, this might be where you have, let's say, pure water 0, and it will move in from a higher water potential to a lower water potential inside the cell. That will cause the cell to burst if it's an animal cell, or lyse or hemolyze, and plant cells will become turgid because the cell wall made of cellulose can withstand the high pressures, and it will fill up with as much water as it possibly can. And again, just another examples of that you can see over and over again, very popular question. Now you might be asked to do this practical, and effectively this practical shows you what would happen if you measured water change in mass against against concentration. So if I did the following concentrations, 0, 0.2, 0.4, 0.6, 0.8 molars of sucrose solution, let's say, as I increased my concentration, the mass change in my potato would go down. And that's because if it was in 0 concentration, it would be in a hypotonic solution. So the water would move from a higher water potential outside the potato to a lower water potential inside the potato. Then as I go down, that gradient, that osmotic gradient decreases, so the change in mass will drop. And the important bit is when we get to here, and this is how you'd work out the concentration of sucrose in your potato. So the point where there is 0% change in mass means there's no osmosis, which means it must be in an isotonic solution. Again, we will look at that in more detail in a practical video at some point soon. Okay, guys, I hope that's been useful, and I'll see you again soon.